Something built like a bear but hunting like a cat is crouched in dense brush at the edge of a tar seep in what is now Los Angeles, watching a herd of ancient bison move past, close enough to smell them but patient enough to wait for the right moment. Its forelimbs are disproportionately massive, built for grappling and pinning prey in close combat, not for chasing anything down over open distance the way a modern lion or cheetah would. When it finally strikes, its jaw opens to nearly 120 degrees, almost double what a modern lion can manage, driving two canine teeth up to 7 inches long into the throat of an animal many times its own weight, delivering a single precise, fatal bite rather than a prolonged struggle. For roughly 2.5 million years, Smilodon was one of the most successful large predators the Americas had ever produced, ranging from what is now Canada down to the southern tip of South America. Then, around 10,000 years ago, it disappeared, alongside almost every other large mammal on the continent. What it left behind was one of the richest fossil collections of any prehistoric animal on Earth. And for more than a decade, the DNA extracted from that collection told scientists something about this animal that turned out to be completely wrong. Start with the name, because it matters. Smilodon is commonly called the saber-toothed tiger, and it is not a tiger, and it was never closely related to one. Tigers belong to the group containing all of today's big roaring cats, including lions, leopards, and jaguars. Smilodon belongs to an entirely separate, extinct branch of the cat family tree, a lineage that split off from the ancestors of every living cat species roughly 20 million years ago, long before tigers, lions, or house cats existed in anything resembling their modern form. The genus itself was formally named in 1842 by Danish naturalist Peter Lund, based on fossils recovered from limestone caves in Brazil, initially mistaken for the remains of a hyena before Lund recognized them as belonging to a cat-like predator. The North American species, Smilodon fatalis, was described separately by American paleontologist Joseph Leedy in 1869 based on a jaw fragment discovered in Texas. By the time modern genetics entered the picture more than a century later, the animal's actual family tree was still an open question, and the first attempt to answer it with DNA got the answer backward. If you're finding this interesting, be sure to subscribe to understand more DNA from the past. Unlike some of the most famous extinct animals in paleontology, Smilodon does not have a DNA preservation problem in the way you might expect. Genetic material has a hard ceiling of roughly 1 to 1.5 million years under the best possible conditions, and Smilodon went extinct only about 10,000 years ago, comfortably inside that window with room to spare. The real obstacle was never whether usable DNA could exist. It was whether scientists could recover it without contaminating the sample with DNA from something else entirely. A problem that would define this animal's genetic history for the next quarter century and lead an entire generation of paleontologists to believe something about Smilodon that direct comparison to the fossil record itself had never actually suggested. In 1992, a research team led by geneticist Stephen O'Brien published a study in the Proceedings of the National Academy of Sciences describing a genuine technical breakthrough, a method for extracting usable genetic material from 14,000-year-old Smilodon fatalis bones preserved in asphalt at Rancho La Brea, the tar pit site in Los Angeles that has produced more Smilodon fossils than anywhere else on the planet. Extracting any usable DNA from bone this old was itself a significant achievement at the time, years before ancient DNA research had developed the strict contamination controls considered standard today, and the technique itself was genuinely innovative for its era. The team sequenced two gene segments, one mitochondrial gene called 12S ribosomal R and A, and one nuclear gene from the feline immune system from three separate fossil specimens, then compared those sequences against 15 living carnivore species, including 9 members of the cat family and 6 non-felid carnivores used as an outside comparison point. Their conclusion placed Smilodon inside the modern radiation of cats, closely related to the living lineage that eventually produced lions, tigers, and house cats, and distinct from an older, unrelated group of superficially similar predators, sometimes nicknamed the false saber-tooths. For a molecular study of an extinct predator this significant, in an era when ancient DNA research was still new, the finding was treated as a genuine advance, and it shaped how paleontologists discussed Smilodon's ancestry for years afterward, appearing in textbooks and documentaries as settled science. It was wrong. In 2005, a separate research team published findings in the same journal, Current Biology, using different fossil material and different techniques, and arrived at the opposite conclusion. Smilodon belonged to a lineage entirely separate from living cats, not nested within it. That result alone was enough to cast serious doubt on the original 1992 finding, but it did not yet explain how such a clear contradiction had happened in the first place, leaving the field with two irreconcilable answers to the same question. A follow up study the next year examined the discrepancy directly and identified the likely cause of the 1992 result. The original nuclear DNA sample had almost certainly been contaminated at some point during handling or laboratory amplification by genetic material from an ordinary domestic house cat. In the early 1990s, the techniques for detecting and excluding this kind of contamination were far less rigorous than they are today, and a few stray cat cells, introduced at any point from field collection to the lab bench, were apparently enough to steer an entire phylogenetic conclusion in the wrong direction for more than a decade. The animal researchers had been comparing Smilodon to, in the study that first placed it among modern cats, may have quite literally been sitting in the same building the whole time. The correction did not stop there. In 2017, a team led by geneticist Johanna Paimans, publishing again in Current Biology, reconstructed complete mitochondrial genomes from Smilodon populator, the larger South American species, alongside three specimens of Homotherium, a separate genus of saber-toothed cat known as the scimitar cat for its shorter, more finely serrated canines, distinct from Smilodon's longer, blade-like teeth. The results were unambiguous. Smilodon and Homotherium diverged from each other roughly 18 million years ago, and both lineages shared a common ancestor with every living cat species alive today, around 20 million years ago. Piemans put the scale of that divergence in blunt terms afterward. In terms of their mitochondrial DNA, these two extinct saber-toothed cats are more distantly related to each other than a tiger is to a modern house cat. The 1992 result had not just placed Smilodon slightly closer to living cats than it should have been. It had placed an animal separated from modern felines by 20 million years directly inside their family tree. Contamination errors, like the 1992 Smilodon case, are not unique to this one animal. Ancient DNA research in its earliest decades was repeatedly undermined by the same basic problem. A fossil sample, however carefully excavated, spends its entire existence surrounded by modern organisms, and modern DNA is vastly more abundant and far easier to amplify through the PCR process than the degraded, fragmented traces locked inside old bone. A handful of skin cells shed by a researcher during handling, a lab technician's own house cat wandering through a workspace, or bacteria that colonized a specimen centuries after burial can all overwhelm a genuine ancient signal if a laboratory is not rigorously controlling for it. And in the early 1990s, most labs simply were not equipped to catch that kind of error before publication. The field's response over the following decades was to develop dedicated clean room facilities, physically separated from any modern DNA source, negative controls run alongside every single sample to catch contamination before it reaches a conclusion, and statistical methods capable of flagging results that look suspiciously modern rather than authentically ancient. The Smilodon correction stands today as one of the clearest before and after case studies in the field, for exactly why those safeguards became standard practice rather than an optional precaution. The final piece of confirmation came several years later, when researchers sequenced the first draft nuclear genome ever recovered from Smilodon populator from a specimen dated to roughly 13,180 years old, discovered in Chile. That specimen holds the distinction of being the oldest complete Paleo genome ever recovered anywhere in South America, a continent where hot, humid, and chemically unstable soil conditions typically destroy ancient genetic material long before researchers ever get the chance to study it, making most South American fossils genetically unreadable regardless of how well their bones survive. The nuclear genome confirmed the same deep divergence found in the mitochondrial data, roughly 20 million years, and went a step further. Using both simulated and real genomic data, the team tested directly for gene flow between Smilodon and living cat species, the kind of interbreeding that has been documented among several modern cat lineages, including recent evidence of hybridization between lions and leopards in the distant past. They found none, not a trace. 20 million years of separation, and at no point in that span did Smilodon's genetic material ever mix with the lineage leading to any cat alive today. Subscribe to not miss the next one. That final study also solved a separate mystery hiding in plain sight. Despite Rancho holding more than 130,000 individual Smilodon fossil bones, representing over 2,000 separate animals, the single largest collection of this species anywhere on Earth, researchers have concluded that recovering reliable ancient DNA from these asphalt-preserved specimens is not considered feasible using current methods. The tar that made these bones so exceptionally well preserved structurally, protecting delicate skeletal detail for tens of thousands of years, appears to be chemically hostile to the genetic material locked inside them, breaking down the DNA even as it protects the bone itself. The breakthrough nuclear genome had to come from an ordinary skeletal specimen in Chile instead, thousands of miles from the most famous saber-toothed cat site in the world, because the world's largest Smilodon graveyard turned out to be one of the worst places on the planet to actually read its DNA. What emerges from this 25-year correction is a complete rewrite of where Smilodon actually belongs. It was not a big cat that happened to grow oversized canines. It was the last surviving member of an entirely separate branch of the cat family tree, one that split away before lions, tigers, and house cats had even begun to differentiate from one another, and one that never once exchanged genetic material with that branch across 20 million years of coexistence. Its body plan, the massive forelimbs, the extreme gape, the blade-like canines built for a precision throat bite rather than a crushing grip, evolved independently, arriving at a superficially cat-like predator through convergent evolution rather than close kinship. The resemblance to a lion is a coincidence of similar ecological pressure acting on unrelated ancestors, not evidence of a shared recent one. Why the animal disappeared remains less settled than what it actually was. Smilodon went extinct roughly 10,000 years ago, in the same general window as the mammoths, mastodons, and giant ground sloths that made up much of its prey base across the Americas. A 2012 study analyzing microscopic wear patterns on the teeth of specimens from that same tar pit site, comparing damage consistent with desperate bone crunching against damage from normal muscle and soft tissue feeding, found no evidence of increased bone consumption or desperate scavenging in the period immediately before the population collapsed, which argues against a simple story of the species slowly starving to death as prey became scarce and increasingly resorting to whatever scraps remained. The more likely picture, according to researchers who have studied the extinction, involves several factors arriving at once: a warming climate reshaping the landscape at the end of the last ice age, the large herbivores, Smilodon, specialized in hunting declining sharply in number, and newly arrived human hunters adding pressure to an ecosystem already losing its largest animals. No single cause has been proven conclusively, and the debate among paleontologists continues. What is no longer in doubt, after 2.5 decades and at least 4 separate genetic studies, is exactly what kind of animal died out in that collapse. Not a tiger, not a close relative of any cat alive today. A member of an ancient, self contained lineage that built one of the most specialized killing tools in mammalian history, twice as wide a bite as anything hunting alongside it, entirely on its own evolutionary path for 20 million years, right up until it vanished for good. The correction took 25 years, four separate research teams, and a fossil from the one continent notorious for destroying the evidence scientists needed. What survived long enough to answer the question was never the tiger name people had been using for over a century. It was an animal that had been alone on its own branch of the family tree since long before any tiger, lion, or house cat existed to be confused with it.